In this video, we will discuss about the cellular RNA can undergo sugar modification. So far, we have discussed in one of our previous videos that about this glycoRNAs, that is, cell surface RNAs might have these sugar modifications. However, this paper describes about the RNAs with sugar modifications uh, are found in the exosomes. So this could be indications of intercellular RNA communications. And they have also found out that if they inhibit those RNA modifications or those biogenesis, if they inhibit or this biogenesis of exosomes, then this papers has also found out that it will increase the glycoRNAs compositions within the cells. And if the glycosylation get stops, then those exosomes that contains glycoRNAs or exosomes that contains RNA with the glycan structures is been reduced. Those things we will cover in our subsequent videos. For today's video, we will just see whether they are also able to see the glycoRNAs that has been previously reported. So in order to check that, they use this kind of reagent, that is AC4 glycnac, AC4 galnac and AC4 manganese. And after incubating them into the cell culture medium and then incubate it for 24 or 48 hours, and thereafter they isolate RNAs and do this spec for biotin conjugations so that they can be visualized using IR 800 streptavidin dye. And this is the visualizations in the panel B. They can see, and they did the experiment in the HeLa cells and the HEC293 cells. So panel B is mainly about the HeLa cells, where they can see that 28S and 18S signals, that is the RNA signal for this different reagent, that is control, manganese, glycnac, galnase. And then when they check the glycoRNAs using the IR 800 streptavidin dye, they are able to see the glycoRNAs presents only in the AC4 AC4 galnase reagent, only in the samples where AC4 galnase reagent is used. And to minor extents, they are also been found in this manganese, but at a longer exposures. And final figure is the merged figure. And thereafter they wanted to check whether these glycoRNAs are in the larger RNAs or in the smaller RNAs. So they separate these RNAs into different populations. That is, first one is fractionate these RNAs into different populations. First, that is greater than 200 nucleotide, and second, which is less than 200 nucleotide. And they checked it for HEC cells and HeLa cells, and they are able to see that most of these RNAs are maximum of these RNAs are greater than 200 nucleotide, but some of the RNAs are less than 200 nucleotide. When they check the glycoRNA signals, they are able to see that those glycoRNA signals are more predominantly expressed in the fractions that are less than 200 nucleotide compared to the RNAs that are greater than 200 nucleotide. Almost there is negligible signals. And here we can see the merged signals. So this all indicates, this till now it indicates that glycoRNAs are present on the small RNAs, and which is also in line with the previously published literatures. Thereafter, they want to check the stability of these RNAs. So they use different RNase inhibitors or RNase degradations agents, such as RNase A. With RNase A, they do not see any significant degradations of this RNase, neither for total RNase nor for small RNase. When they use RNase I or RNase 1, they are able to see some degradations for both total RNase and small RNase also. But when they use this M nuclease with calcium, they see the complete RNase is degraded, and M nuclease without calcium, they do not see any degradations, and in DNase 1 also they do not see any degradations. So all this indicates that these glycoRNAs that are formed seems to be quite stable and can sustain even the RNase A reagent. Thereafter, they check whether this RNase is 5' glycosylated at the 5' end or not. In order to do that, they use this Ri1 reagent, which can degrade or decap the RNase at 5' end. So when they use this Ri1 reagent, they are able to see that in the presence and absence of this Ri1, yeah, Ri1 is the right name, they do not see any changes in the glycoRNAs. Both. So this indicates that these glycoRNAs are not these glycoRNAs or these RNAs are not glycosylated at the 5' end, but there is an internal glycosylation occurs. So far, this is what they have established from the experiments that are presented here. We will discuss further experiments in our subsequent videos, and the story that they have presented in the abstract. We will also see how they justify their conclusions or how they come up to that conclusions that is mentioned in their abstract. That's all for this video. So thank you for your time.